The stem cell review is presented by Celgene Cellular Therapeutics.
I believe that a neural stem cell is that cell which comes from the neuroectoderm. In other words, once the inner cell mass or the epiblast has started giving rise to nervous system, that cell which exists there, to my mind, is a neural stem cell. It's the cell that can give rise to every single cell type in the nervous system. A single cell can do that, so clonality is part of the definition, and therefore that cell must be able to give rise not only to every cell type in the nervous system, but also to other cells like itself that can do exactly the same thing. Now that's an exceptionally broad definition. It's a very interesting question as to if the source of robust, easy stem cells is somewhat inaccessible for a given patient. How does one get them if one wants to actually do therapies and not simply do science? Well, one of the intriguing observations that has been made in the neural stem cell field, and it may well apply to other areas of the stem cell field, is that it seems as if neural stem cells are tolerated by the immune system of other transplant recipients in a way that was somewhat unexpected. That's not to say that there's total immunotolerance. It just seems, however, that when one does a transplant of a neural stem cell in its most immature stem-like state, that it does not provoke the huge immunorejective response that one would anticipate. In our experience, we have not needed to use any immunosuppression and have not seen any immunorejection when we have taken stem cells and put them either into embryonic or newborn rodents. And in some of our basic science and preclinical experiments, these animals, and they represent a range of diseases, have flourished. In other words, they've been able to preserve motor function when motor function would be lost, have a prolonged lifespan when other circumstances they would die. And we've not needed to immunosuppress these animals. We think it's because the neural stem cells do not bear MHC class II on their surface, and that may allow them to become somewhat more immunotolerated. It may not be quite as easy in the adult brain. However, we've learned some great lessons from the old fetal tissue transplantation field. This goes back to the days when fetal tissue was being transplanted into adult Parkinson's patients. And what was learned was that those patients only needed to be immunosuppressed for a few months at the very beginning. And then they no longer needed immunosuppression. And now so many years have passed that some of these patients have died. And as long as 15 years post-transplantation, we've been able to look at their brains and see that the cells have in fact survived integrated send-out processes. So we think that we may be able to use readily available lines of neural stem cells for a range of patients. I think the first applications will be using stem cells as molecular vehicles. In other words, taking advantage of what we call the chaperone effect. So using the cells to deliver, for example, natural products of the cells, like lysosomal enzymes. This is incredibly important for a whole class of neurodegenerative diseases, particularly in the pediatric population, where these kids die horrible deaths simply because they're missing a single enzyme and it cannot be replaced through the vascular compartment because of the blood-brain barrier. These stem cells now can be placed into just a few of the germinal zones of the brain. They will distribute themselves throughout the brain and make a whole range of these lysosomal enzymes, which are usually secreted and very diffusible, and one only needs a very small amount that often is taken up by the disease cells to restore normal metabolism. I think we've already seen at least one clinical trial where that approach has been used. It was by Stem Cells Inc. for Batten's disease, and there's a whole range of diseases that would fall into this category. The biggest first hurdle is the FDA, and in fact most IRBs will look with great interest as to how the FDA has vetted a particular use for a cellular therapy. And what seems to be paramount in the eyes of the FDA is demonstrating safety. Safety probably has an even more paramount importance than necessarily invariant efficacy. Safety is absolute. The FDA never wants to put even a single cell into a patient that could cause a neoplasm or crowd out normal cells or promote dysfunction or abnormal function. In the particular case of Geron, which interestingly became the very first company to initially get FDA approval for putting a bona fide stem cell product into a disease, particularly a neurologic disease in real patients, the reason why they were placed on clinical hold after they initially received approval is that there were some data that emerged from some of their animals, some of their preclinical data, that was unclear. It was not even clear that it was dangerous. It was not clear that it was an unexpected outcome, but it was a little unusual. It was the formation of cysts, and nobody really knew exactly what that meant. Was it simply a benign expected complication, or not even a complication, just an occurrence that happens after one transplants a lot of cells? Or was it, you know, the tip of the iceberg of something that could be dangerous, if not immediately down the line? Well, until all of those questions are answered, it was felt by the FDA that it was not prudent to move cellular therapy forward, particularly for a disease that is not life-threatening. It's interesting that once one accepts the notion that you're going to use the cell to deliver some molecule as one of its natural products broadly throughout the brain, one can start using one's imagination and imagine that it doesn't need to simply be these kids' diseases, but maybe also adult diseases. So for example, other global diseases are something like Alzheimer's disease, that often is characterized by an immense inflammatory reaction. It's very conceivable that one of the natural products of the stem cells are anti-inflammatory agents. They also make a number of neurotrophic and neuroprotective agents. And I think using the cells to distribute these neuroprotective agents in a somewhat broad fashion will be used, I believe, within the next 5 years for a range of diseases that need neuroprotection. I think ALS, Parkinson's disease, stroke, spinal cord injury. Many of these diseases are quite amenable to having their fibers protected, to having inflammation diminished, and having scar formation blunted. I think using the cells then also to deliver molecules, but in this case molecules that we as experimenters and clinicians put in them, will also be used in the next 5 years. I think using the stem cells to deliver tumor-killing genes to definitely brain tumors, whether they be primary or metastatic, but probably to cancers that are metastatic throughout the body, will be a use of stem cells absolutely within the next 5 years. And then I think another use within the next 5 years will be to use stem cells not so much as agents that one takes and puts into a brain or a spinal cord, but using them as models of a disease in a dish from which drugs are discovered. And then these drugs, I think, will be placed into the nervous system or into the body to have access to the nervous system of patients. I am fairly confident that there will be either discovered drugs that are learned to have neurologic actions, or completely new drugs that have neurologic actions in clinical trials within the next 5 years.
At Celgene Cellular Therapeutics, our proprietary placenta-derived stem cells hold the potential to treat a range of serious diseases. Our unique processes for differentiating and producing treatments from this rich source of stem cells will enable us to deliver these therapies on a scale similar to today's biologic remedies. Proud to be at the forefront of this transformation of modern medicine, we are Celgene Cellular Therapeutics.